It's 2 o'clock on a Friday. If you want to get out of here before the, how many feet of snow are coming tonight, we may want to get started. Hopefully we're down to less than an inch by this time. Thank you for all coming this afternoon. This event actually includes 2 awards today. So my first chore is to introduce Mark Coyne, who's going to present the Doyle Peasley Award.
Sure, Mike. I would have thought it would have been your great pleasure to introduce this award. Periodically, since 1991, the Department of Plant and Soil Sciences has given the Doyle E. Peasley Award to an outstanding graduate student of Plant and Soil Sciences. Doyle Peasley was a longtime member of our department, and this award was created in his memory. The basis of the award is high GRE scores, good grades, research strength, and potential for contributions to U.S. agriculture. The award consists of a small honorarium, just enough to take your fellow students out for pizza and beer, and an engraved plaque with the student's name posted in the department. Last year's winner was Matham Al-Ameri. Our winner this year continues the tradition of our department producing outstanding students who we hope one day will return to visit us as distinguished alumni. In addition to the outstanding academic record, numerous service activities, and recognition by national organizations, here are some comments that our winners nominators gave. A self-starter, a front-wheel type drive type of graduate student, the kind all mentors hope to find. A self-starter, I'm seeing a theme develop in these comments. Motivated to understanding processes and completely making broad connections. A gifted student, intelligent, curious, organized, and dedicated, a remarkable communicator. A science fair judge, a workshop developer for middle school STEM activities, the winner of a UK Cafe Women in Diversity Essay Competition, a Tracy Farmer Sustainability Poster winner, and a 2018 Tri-Society's Future Leader in Science Award. Please help me congratulate the winner of the 2017-2018 Doyle Peasley Award for the Outstanding Graduate Student in Plant and Soil Sciences, Matab Bagzaderha. We shake your hand, you smile.
Ready, set, first one. Ready, set, second one.
Congratulations.
Thank you.
Now it's your turn to turn it over to me.
And now is my great honor and pleasure to turn the podium over to Mike Barrett for our distinguished alumni presentation.
Thank you very much.
Well, not only are my remarks not as prepared as Mark's, my notes are gone, so they'll really be totally unprepared. I'm going to start with maybe the last thing. So I could tell stories about embarrassing moments when my phone went off, but if you don't want to have one of those stories for yourself, there's a good time to turn your phone off. So I can't think of a greater pleasure for a teacher and advisor to see his students succeed. So it is my pleasure, and really this is a pleasant thing to be able to do today is introduce our speaker and the awardee for the outstanding alumnus from the department this year, Dr. Jerry Flint from DuPont Pioneer. Jerry did both of his degrees here. He did his master's with me on a plant-based project with herbicides, and then he worked with Bill Witt for his PhD, working on a soil herbicide interaction project. He left here and he went to work for Monsanto. He really started as a field development person, either by luck or by fortune. He started with Monsanto really at the beginning of the whole biotechnology Roundup Ready system introduction and development. He put out some of the first trials testing those lines, the soybean, corn, and cotton lines. And really, he took that experience and over his career of about 20 years with Monsanto, moved from really a science area to an area of regulation, regulatory affairs. And partly because we talked about, he saw that it wasn't just about science, about making these things happen. It wasn't just about science, about delivering these kind of things to growers, but it was also regulation, and it also was the politics and the public policy associated with those registrations that were involved. And so really he took that on and moved up in Monsanto in that area. And about 7 years ago, actually, he moved from Monsanto to Pioneer to DuPont, and really now has become a vice president in the area of regulatory affairs for DuPont Pioneer. Over that time, he's been recognized not just in the industry. If you look at his CV online, you see many awards for his activities, but also he's been called upon to work on really very high level policy panels and at the federal level as far as trade, interactions with China as far as trade and biotechnology. So really, he's from, I would say, humble beginnings. Really a skinny farm kid pretty much. He came here from Purdue. Really has gone a long way and really is a credit to the department. Since this really is about showing graduate students and interacting with some of these alumni about how their careers have developed, I want to go back and tell at least 2 stories about when he was here as a graduate student. And I think basically what they demonstrate is the importance of perseverance in your graduate career and in your life, really. So the first thing was, Jerry was working on a project with me that involved field bindweed. And field bindweed is a weed that takes really a long time to get plant material grown. You grow it up in sticks and pots, and eventually you get this plant that you can do something with. Well, if we have a thunderstorm and it hails and all the glass comes crashing down in the greenhouse, you are set back quite a bit on your bindweed project. I think Gene remembers walking into the greenhouse with Jerry and just was complete disaster, but he recovered from that and did well. The other thing was really more personal. Jerry was involved in really a horrendous car accident and really had a lot of personal recovery to do from that. But he did. He persevered and really did a really fantastic job in both those projects here. Because he was my first student here, he also has the honor of probably having the fattest MS thesis of all time. So I'm sure that prepared him for the rest of his career. So without further comments, Jerry, please come up and give us your story. Thank you.
All right, I tend to walk around, so I'm going to do the mic. Can you guys hear me okay? Yep. Very good. Thanks for that introduction. First of all, thank you. This is quite an honor. When Mike first contacted me about putting together an application, and we did that, when I got the call that I actually was going to be honored with this award, it was quite gratifying. So thank you for those who are involved in the selection and helping put that together. That was really a nice thing. So thank you for that. And also thanks for the great start. We're going to kind of get into it here, but I want to kind of first give you a bit of a background of my career and what I've done. I'm going to talk about some things I've been fortunate enough to be involved in and work on. And then I'm going to leave enough time for question and answer. So, start thinking of them now. We had a couple of roundtables this morning with graduate students, and I was pretty impressed with the thoughtful questions that I received. So keep those coming. So we're going to start with this slide. So I've experienced a lot of change over the last couple years when the company that I was with, DuPont, decided to merge with another company called Dow. And so you'll see Dow DuPont up there. And we're just at the beginning and kind of in the middle of that merger where the two companies, Dow and DuPont, have come together. And they're going to split the combination of that company into three first divisions, which will then become individual companies. So in June of next year, we'll introduce CORTEVA AgriScience as a pure play agriculture company. And so it'll contain the seed brands that we sell, which would include Pioneer, Mycogen, and others. And then the crop protection products. So those will be part of the ag division that will become CORTEVA. So we're starting to refer to CORTEVA now as a division of Dow DuPont, but June of next year, it'll be its own independent company and a pure play agriculture company. And so that's the purpose of this slide, is to remind me to tell you about that, because it's something that if you're involved in the industry, this merger is changing our company, and ultimately that does have an impact on agriculture. But we're excited about it because it does bring together two legacy agriculture companies now focused on a single mission of bringing new products and technologies to farmers. And that's the basis for my title. When I learned that I was going to be able to give a seminar and started thinking about what I would talk about, I wanted to bring this forward. And that's really what's driven me in my career, is to try to bring new products, new technologies to farmers that would improve their productivity, maybe improve their livelihoods. But that's what has driven me in my career, is driving science to solutions. Because you do some work, and I remember Mike and Bill would, as graduate students, would ask us to go out and travel with the extension folks, and that would have been Jim Heron at the time. And Jim would really ask us as graduate students to stand up and explain to a group of farmers in West Kentucky or East Kentucky what we were working on. And his motive for that was, if they can understand what you're working on, you really need to reflect on that, because ultimately what you're doing needs to get in the hands of the farmer. And that has sort of resonated with me throughout my career. And so that's why I chose the title "Driving Science to Solutions," because ultimately that's what we're all trying to do. We're trying to improve agriculture. We're trying to improve livelihoods of farmers around the world. So my career at a glance, I've kind of talked about the first piece of it, but really to improve livelihoods, improve rural communities. Mike indicated some of this, but I did my undergraduate degree at Purdue, a master's and PhD here. And I started out in product development research in the field. And then I moved to Des Moines, Iowa for the first time. So I started my career in Decatur, Illinois in a field development program. And then I moved to Des Moines, Iowa for a regional job. And my role at that point in time was to take those line selections that we had done for Roundup Ready soybeans, Yield Guard corn, and Roundup Ready cotton, and eventually Roundup Ready corn, and help put together the field research program, which helped under pretty high standard of permitting with USDA and EPA in order to bring those through the regulatory system. And that was my first exposure to what operating in a regulatory environment would be about. And that ultimately led me later on in my career to become more of a full-time regulatory professional. When we had the chance to launch Roundup Ready soybeans, I became the technical leader when we launched Roundup Ready soybeans in 1996. I was on a number of other launch teams, including Roundup Ready corn, Yield Guard corn, Roundup Ready cotton, Roundup Ready alfalfa, Roundup Ready canola. There's a trend there, right? And then what that took me to do is, at that time, Monsanto was acquiring seed companies. And so I felt like I needed to learn a little bit about seed. If we were becoming a seed company, I needed to learn about seed systems. And fortunately, I'd taken Dr. Ticroni's class in seed science, so I knew a little bit about seed, but I really didn't know what it took to manage a seed business. So I became a commercial leader in alfalfa, canola, sorghum, and sunflower, which was an eye-opener for me because that really then taught me a lot about the seed industry. So I kind of had to retrain myself in some new concepts. I then did field development and regulatory in Asia Pacific. And so my wife, Dana, raise your hand in the front row. My wife, Dana. I came home one day and I said, we've got a great opportunity. We get to go to a different country, and we're gonna, if we're willing to do that, we can move to Singapore, and we're gonna do that for 3 years, and then I can come back. And so I really didn't know how Dana would respond to that, because we're gonna take our 12-year-old daughter and we're gonna move to Singapore, and it turned out that was a fantastic personal and professional experience for us as a family. But what I did there is we were hoping we could launch that first generation of biotech products into Asia Pacific. And so I was spending a lot of time in China, India, Southeast Asia, Indonesia, Philippines, Vietnam, Thailand. Yep, Thailand is the one I forgot. Occasionally I'd be able to go to Australia and New Zealand, but that was not really mainstream agriculture as far as the rest of Asia Pacific was concerned, but did that for 3 years and then came back. And then that's really what accelerated my career into getting closer to regulators and regulation, because I saw that as the last hurdle. We would do all this great research, we'd do all this great work, we'd submit our regulatory packages, and then we'd wait for the approval. And sometimes those approvals would come in a timely manner, and sometimes they wouldn't. Most of the time they would be based on sound scientific decisions, sometimes they wouldn't. And so because that was a last hurdle to get the technology in the hands of growers, that really interested me. So I started really focusing my career on regulatory. And so that brings me to where I stand here today. And with the new company, I'll have a chance to lead our global public policy initiatives, agriculture development, and sustainability. And that really is about trying to create a freedom to operate for our businesses around the world. And one of the things that I'm really focused on right now is understanding food security and what that means and how that influences public policy in agriculture. And that's not only applicable here in the U.S., but it's applicable around the world. So that's what I'm doing now. And the one thing that when I talked to the student groups this morning, the one thing that I kept getting questions about, what did I learn, and how did University of Kentucky help me with that? One, in my career, I've been a continuous learner, and I really learned that here. I learned how to learn, which sounds kind of odd, but it's one thing to teach, it's one thing to learn, but it's the other thing to apply that learning and then learn how to learn for the next thing you don't even know is coming. When I was in graduate school, there were some folks working on, Dr. Collins and Dr. Hildebrand working on genetic transformation of tobacco and some other crops, but really that wasn't something we could touch and feel yet. So I never really even thought I would even work on that in my career. And it turns out that I did. And so I had to learn how to learn. And the example that I said about learning the seed business, when I started with Monsanto, I didn't know that Monsanto would then acquire seed companies. And so that really forced me to think about my long-term career and what that would mean. And so, developing new skills, adapting and changing. And that's what this University of Kentucky, my experience here, helped me do, is to learn how to learn and to continue to challenge myself in those areas. Because again, you look out in your career 10, 15, 20 years from now, we're going to be working on things that none of us can really imagine. And that's exciting. That's what excites me about agriculture. So let's talk a little bit about some of the things I've been able to work on. So, and how I've been able to leverage that in my career. But, one of the things I learned here is that, and University of Kentucky, I think, still does this, is really drive that multidisciplined approach. And it's not easy. When you think about driving science to solutions and ultimately in the hands of growers, a lot of things have to play out in order to make that happen. It's not just about soil science or crop physiology or better seed varieties and hybrids. A lot of things have to come together to make that grower more productive than they are today. Well, so conservation tillage was one of the first things I worked on. That's really why Monsanto hired me. We were at the point of trying to drive Roundup, the product, into conservation tillage systems. And that required some knowledge of soils and crops and fertility, equipment, and all those things were going on in Kentucky. A lot of it was pioneered here. A lot of it was pioneered by some of the research that Kentucky had done. And some of the concepts that we were talking about in the 80s and 90s about water quality, carbon sequestration, nutrient efficiency, those are concepts that have been around for a while. Now we're applying those to more sustainable systems. And that's part of what my role is today, is to try to think about production systems and how we make them more sustainable. Because of my knowledge and what I gained at Kentucky and in the early part of my career, that helped me leverage that learning into Roundup Ready crops, because Roundup Ready crops became an enabler for further adoption of conservation tillage. So when you go back and look at the adoption rate of Roundup Ready crops and the adoption rate of conservation tillage, because we could have a better solution for some perennial weed control problems, that really did try to help farmers drive those solutions into their operations. And that was the one thing that Roundup Ready crops did. It provided better weed control and crop safety, but you had to deliver those new genetics. And so that's where that multidiscipline approach really starts to come together. It's not just about the trait and the herbicide, it's about the production system and how it's integrated into that. The thing that's exciting about today, I was just talking to Mike and Bill this week about the clover project that Kentucky's working on, and the tolerance to 2,4-D. So new weed control systems are being introduced as the next wave of technology. And I think that's going to be an exciting area as well, is to figure out how those new weed control systems are adopted and developed. Then the third kind of wave of things that I've been able to work on is the next generation of breeding techniques. And it's right on the cusp of solving some of the most challenging things that we've been able to face, some of the more difficult things in terms of agronomic, plant disease, and even some nutritional aspects of crops. And that's a technology called CRISPR-Cas, and I'll get into that in a little more detail in a second. But that's the bottom line, is working to solve big challenges also requires public policy and scientific outreach. And so when I went through all of those, one of the things that we've had to do in each of those is to demonstrate how each of those would solve a public policy area or address a topic that governments were interested in. And so that's a common theme throughout new technology adoption as well. And it's not always easy. So I'm going to switch gears a little bit and talk about CRISPR-Cas. Some of you are familiar with that. When I talked to the graduate students this morning, I was amazed at how much you guys already know about CRISPR-Cas and some of the tools that it can do. I'm not going to read through all this, but this is a reminder for me to talk a little bit about the technology and how it applies to DuPont Pioneer. But CRISPR is clustered, regularly interspersed, short palindromic repeats. What is that? Well, it's actually based on a natural system. It's how viruses actually are incorporated into plants, and plants can become resistant to viruses or other biological systems. So it's based on a natural occurring system. The application to plants involves advanced breeding techniques in terms of what are the changes that we want to make. So with the Cas9 system, we can more efficiently target those changes that we want to make. So it's a new breeding tool that I think is going to find some utility. Again, it's very cutting edge. It's right on the cusp. No one's introduced a product yet in the agriculture, in the plant biotech or the plant area that is based on CRISPR-Cas. We've introduced some things that are based on biotechnology systems, but there's not a product yet that's based on the CRISPR-Cas system. So plant breeding. I like to use this slide to talk about what we can do with breeding. So when you think about a plant like wild mustard, out of wild mustard, we get several crops that we're pretty familiar with today, including kohlrabi, kale, broccoli, Brussels sprouts, cabbage, and cauliflower. And those are relatively new crops based on breeding and selection in the classical way. And so, you know, the breeders have selected characteristics over time to develop those crops. And that's the promise of some of the new breeding techniques that we have, is to be able to further enhance and help breeders make some of the changes that they would like to make that would typically take a long period of time. So how does CRISPR-Cas work? Well, it enables targeted DNA breaks, and it requires a couple things. One, it requires the Cas9 nuclease, which is the DNA cutting enzyme that actually goes in and finds the targeted sequence. It uses a guide RNA to make the changes, the desirable changes that we want to make, and then we utilize that as a way to make sure that we've made the changes that we want to make in the desired way. It requires a lot of the new technologies and a lot of technologies working together to make that happen. So what are some examples of how we can use CRISPR-Cas? Well, one, there's a delete. So then the first sentence, using the word editor as an analogy, you know, you could take a delete where the plant does not have desired output, delete the not, and the plant does have desired output. We can utilize the technology to edit. So in this case, the plant has tolerance to drought, and we can edit that. The plant has high tolerance to drought. We can do a search and replace. And in this case, the example is the plant is susceptible to disease. And the second sentence, the plant is now resistant to disease. So those are some just high-level examples of how the tools can be used in a way to make some of the changes from a breeding perspective that we would like to do that would typically take many, many generations for a breeder to make those kind of selections in improved varieties. And so that's the promise of CRISPR-Cas. So how does that work? So in this example, and we have conventional breeding and back crossing, breeding and selection, and trying to get to the desired traits. With CRISPR-Cas enabled breeding, we can generally take several generations off of that breeding cycle. And so we're able to do it in a very targeted way, a very efficient way to get the desired outcome that we want. And again, it sounds pretty simple. It's very difficult to do. Is anyone working on CRISPR-Cas or gene editing technology? Yeah. So you know, that's a pretty simplistic view of what can go on. There's a lot that has to happen to do that. So what are some of the technologies that need to come together to enable that? Well, first you have to understand elite genetics. We have to have high quality DNA sequencing, which we know we can do now. We have to have informatics and tools and infrastructure to be able to do that, so to make the comparisons that we want to make. And then once we find that out, then it's how do we deliver that in elite genetics? So again, with the analogy that I had before around the breeding, we want to take an elite inbred line or elite variety. We want to make some very specific changes, maintaining all the other agronomic characteristics that we have to make a pretty specific change. And these tools can allow us to do that. And then advancing CRISPR-Cas technology, we're looking for Cas enzymes and systems that can make those edits in a very efficient and predictable way. And so those are the kinds of skills, and again, a multidisciplinary approach required to deliver those products. And at the end of the day, we then use traditional plant breeding as a way to then further those genetics and improve on the elite genetics that we already have. So a lot of core competencies need to come together to do that. What's always been interesting to me about the University of Kentucky is all of those capabilities exist here. And Kentucky's very fortunate because of that. Some of the pioneering work that Glenn Collins and Dr. Hildebrand did to set that in motion, all those capabilities exist right here on campus. You guys should be excited about that, because not every land-grant institution can say that. And it's an advantage, I think, that Kentucky has. So what are some examples of some of the things, the traits that can be developed using the technology? I picked a few. I'm not going to read them all, but you know, for corn, we can reduce phytate. We can produce waxy starch. Waxy starch is used in industrial applications. You go over to virus resistance, and this has been virus resistance in plants is something that breeders have been trying to do for a long time, but it's very difficult to breed for. And so this is one of those things, I think, that depending on the crop, that there are some native genes that we can use and map, and we know about, that can be incorporated into other crops. You take things like tomatoes. You know, we know that there are institutes working on powdery mildew resistance, seedless tomatoes, high GABA tomatoes, and longer shelf life. Those are all targets that processors tell us that they would like to see. So those are just some examples of what can be done with the technology. Now, I don't want you to walk away thinking that CORTEVA, DuPont Pioneer, is working on all those. We're not. Those are examples in the public domain that either universities might be working on or other research institutes might be working on. But this is designed to give you an idea of the breadth of the kinds of applications that we can have. In our program, we're focused more on corn and soybeans at this point. But the message here is these tools can be applied across the different crops that you can imagine. So with that, I'm going to do a few thank yous, and I'm going to do one more slide at the end, but that's just a brief overview of kind of where I've been in my career, some of the things I've been fortunate enough to work on, and hopefully that gives you an idea of some questions that you can ask. But before I do that, there's a number of people I need to thank. I mentioned a few. Mike Barrett. Adele, you might remember this. You were pregnant with your oldest when I sat down in Mike's office. I said, are you sure you're not ready for a graduate student yet? I really want to come to the University of Kentucky and work with you. And he said yes. So so thank you, Adele, for that, and Mike as well. And then obviously, Bill and Mary Witt, through my PhD program, just the work that you did to help prepare me for industry was just phenomenal. So thank you for that. Charlie Slack and Gene in the back. Gene taught me how to work in a lab environment. If you've ever spent time with Gene in a lab, the guy is like a dynamo. I mean, it's amazing how many things that Gene can keep going at the same time. I still don't know how you do it. I understand you're still on that path today, which is phenomenal. But back when I was a graduate student, at one point in time, we had 7 of us crammed into a lab trying to all do our stuff, and Gene would just keep us organized. So, and then Charlie Slack, the field program and learning how to put out field trials. That was when I went into industry. I was one of the few on my class of field development reps that really knew how to put a field program together, and that was Charlie Slack. So thank you for that. A number of other folks in the room that I've worked with over the years, but really thank you for the time that you spent with me as a graduate student and to prepare me for my career. The other person I need to thank is Dana. Dana, stand up. Oh, let's give Dana a hand. Dana has shared my sense of adventure. So I told the Singapore story, but that's not the only adventure story that we can tell. But it's been a great partnership, and Dana actually worked at the medical center and when I was a graduate student, which allowed us to have a little bit better lifestyle than some of the other graduate students that were there at the time. But so we owe a lot to the University of Kentucky Medical Center and the staff here. So thank you for that. And I really do appreciate Dana being open to new ideas and new experiences. So and we have a beautiful daughter. She's 25 years old. She just got married in October. You guys they know me well and my sense of humor. She shares my sense of humor, which is good. Sometimes Dana doesn't quite get our sense of humor, but we deal with that. But she just got married in October, finishing law school. and she'll be entering her professional career here once she passes the bar. So again, thank you very much. I really do appreciate the honor, and I have a few resources for you. I covered a lot in here, but if you want to learn more about some of the areas I talked about, those are some websites you can go to. A couple of them are, the CORTEVA one is about our new company. The CRISPR-Cas is our view of CRISPR-Cas. Seeding Innovation is an industry website as well as GMO Answers. So hit those websites, you'll get a lot more information. And those are just some resources that I pulled together to help you with the next level of questions. So with that, what questions do you have? What questions can I answer? Yep.
One question is, so some, at least the first two applications of CRISPR you mentioned, they could be considered cisgenic, and which so far in the U.S. would not be considered GMOs, but in other countries, I actually don't know. So how might that work out in terms of regulatory applications? So here in the U.S., like say in Europe.
Yeah. So the question is, how do the different regulatory systems look at some of the CRISPR-Cas applications? And that's actually a pretty significant part of my job, because just last week, the USDA clarified their position in terms of how they would view at least the first two versions of what I said with the search, the edit and the delete. And they're pretty clear about what they'll regulate as a plant pest and what they won't. Other regulatory systems aren't that clear, and it's still evolving. So that's part of the work that I'm doing. And that really is part of how we can then launch the products, not only in the US, but globally. But so the regulatory system is still kind of playing out, and we still have some work to do. Because what's driving our thinking is the science is pretty clear. We just got to figure out a way to make the regulatory systems and the science match. If you remember, the first biotech regulations were developed in 1987. It was 10 years before the first biotech crop was successfully introduced. So it does take a while for the regulatory systems to catch up with the products and contemplate what those next products might be in the future. So there's a lot of work to do there, and it's part of the job that I have in my new role.
How many scientists in your organization work on that project with CRISPR?
CRISPR? Dedicated scientists, 50-ish, which would include some field folks that are doing some of the advanced testing with a couple of the products that we're doing. So 50-ish around there. Yep. Yep.
Does that work in Wilmington or Des Moines or Indianapolis?
Yeah. So the question is, where does that research occur? So right now, most of that work occurs in Johnston, Iowa.
Pioneer.
Yeah. Pioneer based. Yep. Yep.
Because I'm muting you guys. We can hear your questions now for the YouTube channel.
All right. Very good. Other questions?
So two additional questions. One, the adoption of no-till agriculture seems to be much less than science would indicate. You might have some comments on that. And is there anything from your company in the pipeline that might be the next round of?
All right, so two different questions. The first one is the adoption of conservation tillage. And I'll get back to one of the comments that I made, is when you look at the first wave of adoption of conservation tillage, two things really came together to get us where we are today in the U.S. I'm going to talk about Brazil in a second. But in the U.S., the farm bills that were put in place at the time had a lot of conservation titles. So from a policy perspective, we were trying to encourage farmers to adopt conservation tillage systems. Then the introduction of Roundup Ready crops actually helped facilitate that. So a technology piece and a public policy piece started to work together to bring farmers to adopt those technologies. Now, the farm program started to go away. the technologies were adopted, and so then it became economic drivers in terms of what would be the next economic driver to encourage growers to adopt the technology without those two other pieces in place. You look at a country like Brazil, almost every acre has some sort of conservation tillage, and about 60% of the corn and soybean acres are no-till. That tells you that really there are very few technical limitations in the U.S. to be able to do that. We need to help drive the adoption at a much more rapid rate in the U.S. And I think with some of the emphasis on sustainability and sustainability of farming operations and how agriculture works with public policy, I think we'll start to see additional funding in the areas of conservation tillage adoption, whether it's no-till or reduced tillage. But it has plateaued in the U.S. It hasn't increased for quite a few years. So we're going to do something to drive it to that next level. The other one is there's something in the pipeline that's similar to Roundup. Roundup is pretty unique, and it will be very difficult to find something that has the same environmental profile, the same health and safety profile, and the same utility across the weed control spectrum. It's a very, very difficult, and companies have been trying to replicate the success of that molecule since it was first introduced, Bill, what, 1976, something like that? Yeah. So it's a very difficult, with all those things that come into play, it's very difficult to improve on that. Not to say it won't be done, the hurdle is pretty high. Yeah.
So, based on the adoption, I think farmers are very much in favor of the GMO crops and the technologies. You just highlighted one of the ecological benefits, and that it makes it easier to do conservation tillage or no tillage in their system. I'm still surprised and disappointed that the public in general, there's a lot of backlash against GMO crops. So I guess my question is, do you think that CRISPR will help us get away from that issue, or do we need to develop some sort of end user benefit that the end user, the consumer, actually sees or tastes before we get acceptance?
Yeah. So the question is, John, how do we drive more technology acceptance in some of these areas, right? So with one of the groups this morning, we talked about this a little bit, but I think we've got to take a step back and reflect in terms of how we actually talk about what we do. And one of the things, and I caught myself doing it here today, is that I want to talk about the cool science and how we do things. What we really need to do is flip that conversation and talk about some of the challenges we're trying to solve or some of the problems we're solving, and then talk about the technology that we're utilizing to do that. We have to flip that conversation. And so I did it today. It's not an easy thing to do, because when you're a scientist, you want to talk about the cool science, right? Well, I think we need to think about then, repositioning that as these are the challenges or problems which we're trying to solve. And oh, by the way, this is a technology that we use. We didn't do that with biotechnology. We started with the cool science, we started with the regulatory systems, and then we thought about the general public. And I think doing that again, we're really trying to change that around with the CRISPR technology. Talk about the benefits of the products first. But thank you for the question. Yep.
I'm getting a lot of questions from friends and family. I'm in particular with Roundup, been hearing of human health and environmental concerns. How do you answer that question, just on a lay audience?
Yeah. So the question is, we're getting a lot of questions about the product Roundup in some different areas that we haven't experienced before. It's one of the things that the realization of the environment that we're working in today is there's a lot more awareness in the media and social media around agricultural systems. We've got to tell our story better. We've got to talk about the benefits of the product, the long history of safe use, and the different applications that we've had, because the science would actually indicate that from a human health and environment perspective, there are no issues. Now, what we've got to be able to do is counteract some of the questions that we're getting in terms of challenging the data that exists. But it's something that is endemic on agriculture, and something that we've got to do just in general across the different technologies that we're using, is talk differently about what it means.
So how do you answer someone kind of like an elevator speech when they ask that question? Do you have any specific thing you can say to them?
Yep. I go back to my experiences working on Roundup. I've been working with Roundup over 30 years in different applications. I trust our science. I trust the National Academies of Science. When they went back and looked at their meta-analysis around some of these very questions that we're getting, the National Academy of Science review basically said there are no issues. And so we have to rely on the experts that have worked with and experts in the area with the safety of the product.
So what was wrong with the WHO study a year or two ago that claimed it was carcinogenic? What was missing in their science?
Yeah, so the question is what was missing in the WHO science? It didn't contain the complete data set that the National Academies actually considers. So it was a pretty defined set of studies they looked at, not considering the whole or the weighted evidence approach that you would typically take in a meta-analysis type approach. So the methodology was not as robust as what we've seen in the National Academy studies. Yep.
So how does it feel not to have that big red M hung around your neck in your new job? So Monsanto isn't causing the world's troubles. Is that being a placebo?
So the question is, I spent the first 20 years of my career at Monsanto. So what has it been like? Yeah, it's interesting because for whatever reason, Monsanto has taken a lot of heat with some of the products and technologies they have. For me, I still, as being part of the industry, I have to talk about us all together. And so I'm not afraid to tell folks, who I worked for and what I've done. And so I'm proud of the work that we did, and I'm proud of the industry that I serve. And that's I don't ever hide from the fact that I work with Monsanto. It's unfortunate because there are a lot of good people, a lot of former graduate students that came through this program, and me included, worked within the Monsanto system, and great scientists, good scientists. I think it's unfortunate that a lot of the attention has been played on a company like Monsanto. Yes.
So over the past few years, we've sort of seen a trend of consolidation in the world of agricultural biotechnology. And I know some of the regulatory concerns, perhaps a little bit outside of product safety perhaps, is that this consolidation is going to lead to a stifling of innovation. Would you have anything to say about those concerns?
Yeah, so the question is, what are all mergers and acquisitions going to do, and how is that going to impact agriculture? I was talking to Charlie over lunch. When I was in graduate school, we saw the first wave of consolidation in the ag industry, and that was driven by the farm economy at the time. We've seen other waves go on as well. And what you tend to see is that a company like ours, DuPont, actually in doing that merger, we actually sold some of the DuPont products to another company, FMC, which is going to improve their portfolio. So it essentially sets up another company to potentially be successful. And that's typically what you see in the ag industry is that other competitors will start to emerge. One of the things that we track as a company is the amount of investment in startups, venture capital, and where those investments are occurring. And what we've seen is over the past 3 to 4 years an uptick in some venture capital money going into things like predictive agriculture, digital ag, some startups around biological products. So we're starting to see some of those companies emerge. And that says that there's some investment room left in agriculture. And I've seen that in my career. You see these ebbs and flows within the industry, and ultimately the cycles play out, new products emerge, and new competitors emerge as well.
along those lines. So now Monsanto is what, part of Bayer Crop Sciences, right?
Correct. So Bayer Crop Science is gonna purchase Monsanto. Yep.
They're gonna drop the Monsanto name.
Who knows?
Yeah.
Well, before I ask Rebecca to come over and make a presentation to Jerry, I just want to recognize Dennis Tacroni, because Dennis Tacroni, I think, walked into my office as chair one day and said, I think we need to do this. So really, this goes back to Dennis's idea, and I think he certainly deserves a hand for that. With that, I turn over to Rebecca with
All right, Jerry. Well, thank you very much for coming. It's a pleasure to meet you and listen to your talk today. I know this is investment of your time, and I appreciate that you've come. I thought it was very exciting today to hear and listen to the pride that Jerry has been expressing in our department, in our university. It makes me proud of you all. I like the fact that we have pride in both our faculty and our staff, so I hope this has been a good illustration of how we all work together to produce amazing people and people that we get to celebrate. So you are the 12th annual winner